Hello students, this is Ms.PRIYA SETHURAMAN from the Department of Food Processing Technology from Amet deemed to be university. Today we are going to look on the title on Metabolism of Yeast in Alcoholic Beverages. So what are beverages? So beverages are portable drinks which have the thirst quenching, stimulating and refreshing qualities. So one tends to drink for 6 reasons. For one is they usually quench the thirst. Second one, they get to get drunk. Third one, during the party time they usually drink. Fourth one, they usually drink for thirst. So moving on to the next slide. Beverages are a liquid formulation intended for human consumption. So this is the definition for beverage. It is a portable drink. So first one, the beverage. So the beverage is classified into 2 types. One is non-alcoholic and another one is alcoholic. So under non-alcoholic beverage, so it's a beverage which doesn't contain any alcohol. So these non-alcoholic beverages are classified into 2. One is non-carbonated and another one is carbonated. So whereas in alcoholic it is classified into 2. So one alcoholic drinks can be taken out from fruit or sap, or it can be taken out from the grain. So coming on to non-carbonated drinks. So non-carbonated drinks, they come like fruit juices, fruit drinks, fruit nectars, coffee, tea. So when it comes to coffee and tea, they are stimulating beverages. So they help to stimulate the body. So when we are tired or any worn out, so drinking coffee and tea will help us to shed the tiredness. So the next one, carbonated beverage. In carbonated beverage, soda, cola water and tonic water comes. So in carbonated beverage, the drink is filled with carbon dioxide. So that is carbonated drinks. So moving on to fruit sap. So this fruit sap in under alcoholic beverages, the fruit sap is divided into 2, non-distilled and distilled. So in non-distilled, usually wine corresponds to the non-distilled drink. So wine which is prepared, wine can be prepared from grapes, orange, apple, etc. The next one is distilled. So in distilled beverage like brandy, whiskey, rum, vodka, gin, so everything comes under distilled beverage. So moving on to grains. So in grains, non-distilled beverage, so only grains like rye, oats, rice, wheat, so these are grains from which they can get alcohol. So for example, beer is one thing which is done by using grains, and next one whiskey. So whiskey is also prepared from ingredients like grains. So the title today is on beer. So beer is an alcoholic beverage produced by the saccharification of starch and fermentation of the resulting sugar. So the fermentation happens by the help of yeast. So yeast plays the major role in fermentation, which consumes the sugar which is present in the beer and converts it into alcohol and ethanol and carbon dioxide. So this ethanol is a psychoactive drug. Which is antidepressant drug. So that's why people tend to drink beer to forget all the things. So the next one, due to the saccharification of starch and fermentation of the resulting sugar, we get the beer. So the beer is usually flavored with hops. Hops are a female inflorescent plant. The scientific name of hops is Humulus lupulus. So it corresponds to the flavor, it gives the bitterness to the beer, and also it gives the taste of the beer will get changed when hops are added. So the next one, the strength of the beer. The strength of the beer is classified around 4 to 6% by volume. So when it is 4%, it is standard beer. When it comes to 6%, it is super strong beer. 6 to 10% beer will correspond to the super strong beer, and it comes in various like 650 ml, 350 ml. So the range of the drinks varies. So moving on to the next slide. Okay, so what is in beer? So beer is a drink where all these 5 ingredients are present. So barley, so the barley gives the body and the soul of the beer. Okay. The next one, yeast. So yeast gives the life to the beer. Third one is hops. So hops is the spice which is added inside the beer. The water corresponds to the 92% of water. So beer is made up of 92% of water. So water gives the integrity and purity of the beer. Next one, the final thing is adjuncts. Without these adjuncts, a beer is nothing. So adjuncts like rice, corn, wheat, rye, oats. So these adjuncts are added to the beer in order to give the body and in order to give the taste to the beer. So next slide. So the next slide is barley. So what is barley? So barley is the thing which gives the body and soul to the beer. So barley malt gives the beer the natural enzymes. So at a corresponding temperature, so these barley malt releases out the natural enzymes. So these natural enzymes gets converted to sugar during the fermentation. So next one, it also gives the malty sweet flavor to the beer. So the color of the beer depends on the nature of the barley malt. So next, the foam head. As you see here, the foam head which is formed, that is due to the barley which has the proteins. So barley has so much of proteins which corresponds to the foaming nature. The next one, these barley are of 2 types. One is 2-row barley and 6-row barley. So these 2-row barley and 6-row barley are grown in many countries like Germany, US, then Scotland, etc. The variety of barley malts. So there are variety of barley malts that helps to determine the color, texture and flavor of the beer. And one more thing. So the color of the beer depends on the nature of the barley malt which is used. So the next slide. So the next slide is on the yeast. So yeast is the only microorganism, the unicellular microorganism which gives a life to the beer. So the life of the beer is given by the yeast. So yeast is a unicellular microorganism. When it eats the sugar which is present in the beer, it gets converted to carbon dioxide and alcohol. So there are 2 types of yeast which are produced in beer. One is ale and one is lager. So ale beer are top fermenting yeast, whereas lager beer are bottom fermenting yeast. So moving on to the next slide. So moving on to the next slide, hops. So the hops are the thing which gives a spice to the beer. So mostly female, as I said earlier, in hops only female inflorescence, a part of the flower is used, which corresponds to the flavor as well as bitterness. So the bitterness, the aroma and the foam are given out by the hops. So the microbial stabilization of hops have the antiseptic qualities. So beer has antiseptic qualities. So the aromatic flavor of the beer is produced, it is given by the hops. The bitterness is also contributed by the hops. Second one, lastly, the foam stabilization. The proteinaceous substance are responsible for the foaming nature in beer. So there are 2 types of hop styles. One is aroma hops and the other one is bitter hops. In aroma hops, hops like Sass, Fugel, Halachua are used, and in bitter hops, Brewer's Gold and Unique. So these 2 are bitter hops which are widely used for bitter beers. So the next topic is on the water. So the water gives the integrity and purity to the beer. So as I said, water corresponds to the 92% of beer, and there are two types of water, one is hard water and another one is soft water. In hard water, it helps to give the crispiness and cleanness of the beer. So, the second one is soft water, it adds softness to the beer. Next slide. So, the next slide is on adjuncts. So, as I said, the adjuncts are a thing which should be added in the beer, which gives the body and it gives the thickness to the beer. So, the adjuncts are added in order to change the color, in order to change the flavor, etc. So, then adjuncts like grains, mostly grains are used as adjuncts like maize, rice, wheat, rye, corn. Broken grits of maize are used, and like fruits like raspberry, cherry, strawberry are used, and spices like cinnamon, etc. are used in beer, which corresponds to the adjuncts. So, the next topic is on lager. So, lager means to store. So, the type of yeast which are used to ferment a lager type of beer is a bottom fermenting yeast. So, bottom fermenting yeast are Saccharomyces euverum and Saccharomyces Carlsberg genesis. So, they are usually fermented at lower temperature around 50 degrees centigrade, and mostly these types of beer produced by the lager types of lager are very smooth and they give less fruity aroma to the beer. Moving on to the next slide, ale. So, ale are a top fermenting yeast. So, they are usually fermented at higher temperature around 64 to 72 degrees centigrade, and these ale type of yeast like Saccharomyces cerevisiae are usually done for ale type of yeast. So, moving on to the next slide. So, brewer's yeast. So, brewer's yeast are usually given in bakery products to ferment bread. So, usually they are done to ferment bread, buns, etc. So, these types of yeast are given like Saccharomyces cerevisiae. So, they are pulverized cells of Saccharomyces yeast. They are dried and they are usually produced by budding. So, there are various varieties of yeast which are used. For example, as I said earlier, ale type of yeast are top fermenting type, so that corresponds to Saccharomyces cerevisiae, and the lager is a bottom fermenting yeast, Saccharomyces euverum, which is formerly known as Saccharomyces Carlsberg genesis. So, the next topic is on yeast. So, yeast are eukaryotic single celled microorganism which comes under the fungus kingdom. So, there are 1500 species of yeast which is identified. So, the size of the yeast varies greatly. So, typically they measure between 3 to 4 micrometer in diameter, and sometimes yeast can also come across 40 micrometer in size. So, the next topic is on yeast. So, usually yeast reproduces by asexual reproduction, and sexual reproduction takes place. When it comes to asexual reproduction, mitosis takes place, and the reproduction is done through budding. So, you can see the picture over here which gives out the here this is the mother cell and this is the daughter cell. So, when it starts reproducing, the mother cell produces the daughter cell. When it gets matured, when the daughter cells get matured, what happens? The daughter cells leave the mother cell, leaving a scar behind the mother cell. So, this scar which is left on the mother cell corresponds to the age of the yeast. So, the age of the yeast is determined by the number of scars that is present all over the body of the mother cell. So, the next slide is the main features of a typical yeast cell. So, moving on to the picture here. So, this is the bud. So, here this is the bud which is done by budding. So, here this is the nucleus, and here the endoplasmic reticulum which is near to the nucleus, here mitochondria, the cell wall, the cell membrane, the vacuole, Golgi bodies, the fat globules, the secretory vesicles. So, these are present in a typical yeast cell. So, the shape and the structure of the yeast cell depends on, they may be oval or they may be global in size. So some may be elongated or some may be in special sizes like thing, you can find it over here. So next one, some cells, the movement of the cells are they are done by pseudoflagella, so pseudomycelium. So pseudomycelium corresponds to the movement of the yeast cell. So the next one, the length, width. So the width of the yeast is 1 to 9 micrometer and the length is 2 to 10 micrometer. They are spherical in size and one more thing, they are larger than bacteria. So the spherical cells are like this. So you can see over here, as we saw in the previous slide. So it is just like the budding. So this is the bud and this is the mother cell. So the next thing which we are going to see is the main features of the yeast cell. First one is the cell wall. The cell wall is surrounded by a rigid envelope. So this rigid envelope is made up of two types of carbohydrates. They are glucan and mannan, and glycosylated proteins. So that the glycosylated proteins are a phosphomanoprotein. Second one, so the cell wall determines the shape of the cell. So it also provides a barrier against the osmotic pressure in cytoplasm, and generally it maintains the integrity of the cell. The next one, the cell wall also helps to maintain the shape, and also it helps the other cells inside the body to be inside compactly. So the next thing, it also produces a capsular material which is produced extracellularly. So the function of the cell wall is to selective transport of low molecular weight compounds. So the next thing is the cytoplasm. Cytoplasm is the ground matrix. The cell structures are suspended inside the cytoplasm, and the pentocycle enzymes, glycogen, so these types of enzymes are present inside the cytoplasm. So the next one, inside thing is a nucleus. Inside the nucleus, nucleolus is present, which has 90% of DNA and RNA and polyphosphate compounds. So there are also a number of chromosomes which is also present, and in nuclear membrane a pair of unit membrane with pores is also present, which is around 85 nanometer. And cell division, so that the nucleus divides, and function here inside, which is here, is genetic and hereditary control. So inside the yeast cell, endoplasmic reticulum, which is also present, which is the double membrane system. So ribosomes are present, which are responsible for the protein synthesis. So moving on to the next slide. So the next slide is on vacuoles. So vacuoles are spherical in shape, so they are more transparent, so they vary in size, and they also serve as a food storage of the yeast cell. So the function here is they are usually they autolyze. See, autolyze is nothing but they kill themselves. And mitochondria, so there are mitochondria, Golgi bodies, lipid globules, and so they are also present. So mitochondria, as we all know that mitochondria is a powerhouse of the cell, and which contains DNA, RNA and the enzymes which are responsible for TCA. TCA is a citric acid cycle, and lipid globules are also present. So in lipid globules, it contains only lipids, and lipid does not contain any membrane. So the next slide. So the next slide is on Saccharomyces cerevisiae. So Saccharomyces in Greek it is called as a sugar mold, and cerevisiae in Latin is called as beer. So this Saccharomyces cerevisiae was used as a microorganism, which is a eukaryotic microorganism, which is used to ferment sugar in baking and brewing. So the next thing, so in earlier days they are first isolated from the skin of grapes, and they usually flocks. The flocks are usually formed when fermentation happens. So this flocculation is a process of coming out of solution and clumping together. So these yeast flocks together, and they form a clumps over the beer. So the next slide. So the next slide is on the life cycle of the yeast cell. So here you can watch the diagram here. So the reproduction takes place asexually and sexual reproduction. So here in asexual reproduction, so the bud, this yeast which forms the bud, So look here, the bud is formed, and once the bud grows from the mother cell, it leaves the mother cell and leaves a scar over here, which and it develops individually. So whereas in the sexual reproduction, so the mating pair, these two mate together and they form the diploid cell, and this is how the sexual reproduction takes place. So the next slide. So the next slide is on the nutrition. What does the yeast eat to survive? So the naturally in the yeast there are two types of nutrition which is required. One is macronutrients and another one is micronutrients. So these macronutrients are building blocks needed for the development of the new cell material. The next one is micronutrients. So these micronutrients they are usually facilitate the biochemical reactions. So these two are micro and macronutrients. In micronutrients, trace elements like calcium, magnesium, manganese, zinc, etc. are corresponds to the trace elements which are present in micronutrients. So they are even the requirement of it may be very low, but they are essential for the growth and the development of the organism. Second thing, in macronutrients, so like glucose, fructose, sucrose, so they are macronutrients, so in which helps in the energy source of the organism. So the nitrogen sources like amino acids, ammonia, nucleotide, bases, peptides comes under the nitrogen sources, and for phosphate sources, inorganic phosphate and organic phosphate compounds comes under the phosphate sources. So for sulfur sources, inorganic sulfate, organic sulfur compounds comes under the sulfur sources. So moving on to the micronutrients, some yeast can synthesize their own vitamins. So it like pantothenic acid, biotin and folic acid, niacin, riboflavin. So these acids can be synthesized by the yeast cell itself. So we need not put any other vitamin source, because yeast itself produces these types of vitamins which is good for the body. All these vitamins are present in the molasses. Okay, then moving on to the next slide. So the next slide is on the macronutrient energy sources. So these macronutrient energy sources, they are monosaccharide. Macronutrients are what? Monosaccharide, disaccharide, trisaccharide, pentoses, etc. So in monosaccharides, glucose, fructose, galactose and mannose comes under monosaccharide, and in disaccharides, sucrose, maltose, mellibiose comes under disaccharide, and in trisaccharide, raffinose, the sugar raffinose comes under trisaccharides. So the oxidative substrates which are present in the macronutrient energy sources are pyruvate, acetate, lactate, glycerol and ethanol. So the next slide. So this is the pictorial representation of the yeast which is used for fermentation. So can you see the thing, the budding cells, the budding thing from mother cell. So this is the mother cell and this is the daughter cell, which the reproduction which is done through budding. So this is the microscopic view of the daughter cell and the mother cell. So these yeast which are elongated, these types of yeast which are elongated comes under the wild yeast. So these are wild yeast. So this elongated substances, substrates, these are wild yeast which comes under the thing. So here these are flocculation. So this is what called as flocculation, where the yeast comes together. So the reproduction takes place, and once the daughter cells leaves the mother cell and gets flocculation, and these comes under the flocks. So next slide. So the next slide, the yeast, the growth phase of the yeast. So once the yeast starts consuming the sugar, glucose which is present inside the beer, what happens? The first stage in this is the lag phase. So here this is the lag phase. This is the lag phase where the yeast consumes the sugar which is present and it is in dormant state. So once yeast starts consuming more and more sugar which is present, what happens? It enters into the stage called as log phase. So here log phase, the yeast starts reproducing very much faster, and once when it reaches a stage where it cannot reproduce anything, so the stage is called as stationary phase. So here the stationary phase, they become stationary, so they become stable, they do not eat any, so they do not consume any sugar which is present, and the final stage is the death phase where the yeast starts dying out. So most of the fermentation is done by the stationary phase cells. So here in stationary phase cells, the rate of the growth is equal to the rate of the death. Okay, the next one here, no death, no growth occurs in stationary phase. So the next slide here, how does ethanol inhibit the yeast? So here it displaces the water of hydration and changes the properties of a protein lipid interactions. Second thing, it denatures the proteins, it disrupts the protein active sites, it allows the passage of protons from the medium of cells leading to the acidification of cytoplasm, and it removes the protons which requires the expenditure of energy. So the next slide here, there are 9 characteristics of better yeast for alcoholic fermentation. So the first one, the yeast should have high alcohol tolerance, so it should be above 12%, and the second one, the high ethanol yeast and productivity. Third one, tolerance, it should be tolerant to the inhibitors. Fourth one, it should have a minimum nutrition supplementation, it should have high sugar tolerance, it should have high temperature tolerance, it should be above 40 degrees centigrade, and it should be like a flocculating type. It should not be a non-flocculent yeast, it should be a flocculent type of yeast, and it should produce less products during the fermentation process, and that lastly the broad substrate utilization must be in a required range. So the next thing here, what happens to the sugar when the yeast starts consuming? The fate of the sugar during the Saccharomyces cerevisiae metabolism. Here these are the fermentable sugars, like as I told, they are macronutrients, glucose, sucrose, fructose, maltose, maltotriose. So they are fermentable sugars which undergoes the glycolysis pathway. So in glycolysis pathway, the sugar gets converted to pyruvate. So here once after glycolysis, the pyruvate is the end product which is formed in glycolysis process, and here this pyruvate undergoes fermentation and respiration stage. In fermentation, it lets out ethanol, carbon dioxide and 2 molecules of ATP. ATP is adenosine triphosphate, and in respiration, carbon dioxide, water and 38 types of ATP molecules are released out in respiration. The next topic here is the alcoholic fermentation process. So it is a glycolysis pathway. So here glucose gets converted to pyruvate, and then naturally after pyruvate it undergoes the citric acid cycle, and which let out acetaldehyde, and then it gives out ethanol. So here in glycolysis, 2 molecules of ATP is produced. And the next slide. So this is the alcoholic fermentation, that is the glycolysis. So glycolysis is the most important thing which is formed during for the formation of ethanol. So here the glucose is converted, it undergoes the various stages and gets ultimately here the pyruvate is formed. So after pyruvate, once the pyruvate is formed, it undergoes a reaction where acetaldehyde is formed, and this acetaldehyde let out carbon dioxide and ethanol is released naturally. Next thing. So the next slide here, we are going to talk about the process utilization by yeast to produce the ATP in the absence of oxygen, that is the anaerobic absence of oxygen. And the glycolysis here, in glycolysis 2 molecules of ATP is produced and 2 molecules of pyruvate is released, and so here in 2 molecules of pyruvate they are oxidized to produce 2 molecules of carbon dioxide, and ethanol is the final product which is formed in the glycolysis. So this is the previous picture which I have put here for the clear explanation. So here the glucose which undergoes to pyruvate, and then ethanols, 2 molecules of ethanol will be formed. So, this is the glycolysis phase. So, the next slide. So, the next one here, HMP. What is HMP? HMP is the hexose monophosphate shunt pathway or the pentose phosphate pathway. It is otherwise called as Warburg-Dickinson cycle. So, this Warburg-Dickinson cycle here, as I said, in glycolysis, glucose, here glucose gets converted to various stages, and here glycerol 3 phosphate and xylulose 5 phosphate is released. So, this hexose monophosphate shunt pathway is otherwise called as pentose phosphate pathway, which is also responsible for the alcoholic fermentation. So, these are the fundamental things for the development and the growth of the yeast cell. So, here in aerobic and anaerobic. So, in aerobic thing, glycolytic pathway comes, that is EMP pathway, the Emden-Meyer-of pathway, and in anaerobic TCA cycle, that is oxidative phosphorylation takes place. The thing which I have discussed earlier. So, the next thing here, 2 molecules of ATP is produced, that is in glycolytic pathway 2 molecule of ATP is produced, whereas in anaerobic, that is in TCA cycle, 36 molecules of ATP is produced. So, here in this it has a weak cell membrane, whereas in anaerobic thing they have the cell membrane with UFA and sterols. So, sterols are nothing but nicotinic acid, and whereas in aerobic thing it takes place through fermentation, and in anaerobic it is alcohol tolerance and respiration. So, the next slide which we are going to see on is the metabolic roots. So, metabolic roots which are taken by the brewer's yeast to produce the flavor compounds. So, once sugar gets fermented by the yeast, it releases out the ethanol and carbon dioxide. So, in between there are number of flavor compounds which are released by the yeast, like vinyl diketones, fusyl alcohols, esters. So, these are some of the flavor which is released by the alcohols. They come under higher alcohols. So, the fusyl alcohols are otherwise called as higher alcohols. So, this vinyl diketones. So, this vinyl diketones travels from the malting stage itself. So, the next slide is, so the next thing, the beer fermentation flavors. So, inside wort. So, what is wort? Wort is the heart of the fermentation. So, wort is the heart of the brewing. It is otherwise called as the heart of the brewing. Here the wort, inside the wort there are sugars, amino acids, etc., which are present inside the wort. So, wort is a very pure liquid where bacteria or wild yeast cannot survive. So inside wort, so this is the organism, that is the yeast, the unicellular microorganism. Here glycerol, higher alcohols, esters, fatty acids, sulfur compounds, ketones, aldehydes, phenols, and once yeast starts consuming all the products and it gets released, it releases the ethanol and carbon dioxide. What happens to the yeast? What is the fate of the yeast? So, what happens? The yeast dies. So, it lyses nothing but it kills itself. Autolysis takes place. So, this autolysis, what happens? It forms the, it gives the form, it dies and gives the foaming nature of the thing. So, the foam will be formed once the yeast dies. So these are the phenolic compounds, glycerol, alcohols. So, in wort, since wort is a very pure liquid and it doesn't have the load of bacteria or wild yeast, because it is it is in purest form. The next slide is, so the ester production of these, over 90 distinct types of esters are produced by the yeast in beer. So, so the yeast may also produce flowery aroma, spicy aroma, flowery aroma, fruity aromas are also produced by the yeast. So, these are the important esters, that is isoamyl acetate, ethyl acetate, isobutyl acetate, and ethyl carpurate, which are also produced by the yeast. So, these are the important esters which are produced by the yeast. The next slide. So, the next one is the glycerol production by the yeast. The glycerol is the quantitatively one of the most important products during the yeast fermentation, and this glycerol corresponds to the viscosity and the body of the beer. The next slide is the higher alcohols. As I said, these higher alcohols are otherwise called as fusyl alcohols, fusyl oils. So, they are increased and reduced. So, this is nothing but they are increased by the high fermentation temperature, and during the fermentation, during the mixing, they get increased, and if the concentration of the amino acid is less, the air alcohol gets decreased inside the wort, and if there is a more air inside the thing, the air alcohols will be produced, and if they can be increased if they are having higher gravity. So, whereas in higher alcohols can be reduced by these factors, like if the yeast has higher pitching rate, these higher alcohols can be reduced, and if the pressure during the fermentation gets reduced, higher alcohol will be produced, and if the oxygen is not there after pitching, higher alcohols will be reduced, and if enough amount of amino acids are produced, these higher alcohols will be reduced. So, there are 80% of higher alcohols are built up during the fermentation. So, they can't be influenced, so they are not responsible for any maturation phase of the fermentation process, so brewing process. So, the next slide is the higher alcohols. So, it gives they are classified, these higher alcohols are classified into three. One is aliphatic alcohols, aromatic alcohols, and multivalent alcohols. In aliphatic alcohols, these are the four types of aliphatic alcohols which are produced by the yeast. So, the next one, they are aromatic alcohols, and these tyrosol, tryptophol, 2-phenyl ethanol, so they come under aromatic alcohols, and multivalent alcohols, glycerol, 2,3-butadiol is produced under the higher alcohols. So, the next slide here, the autolysis. As I told, what happens to the yeast after fermentation, what is the fate of the yeast. So, what happens? So, it gives some meat like flavor and aromas, and it gives harshness to the beer. So, here the viability of the yeast gets reduced. Once autolysis is the terminology which describes that the death of the cells, it kills itself. So, autolysis is nothing but killing the cell by own. So, the next, it gives the meat, like flavors and aromas, and it gives a harsh bitterness. Bitterness is corresponded by the hops, but if harsh bitterness is a taste during the beer drinking, it is ultimately it is due to the yeast autolysis. Okay. The next thing here, if the viability of the yeast get reduced, it produces the harsh bitterness to the beer. If yeast releases compounds like proteases, what happens? The meat like aromas will be produced, and if the pH is increased, what happens? The yeast gives the meat like aromas, and the bitterness will also be very harsh. The next thing, it can be increased by the length of the time. How long does the beer, the yeast survives inside the fermentation? It also the autolysis it is determined by the length of the time the yeast sits on the yeast. Beer sits on the yeast and the temperature of the yeast. Next thing, it can be reduced by removing the yeast. Once it has fermented the ethanol, once it has fermented the sugar into ethanol and carbon dioxide, the yeast should be taken out. It should be removed completely. So, if it is not removed, what happens? The death of the yeast cells will be more, and harsh taste and harsh bitterness will be produced by the yeast. So, these are some of the sulfur aromas will also be produced by the yeast. So, it can be increased by the following factors, like if the wort has less oxygen. So, once the wort gets less oxygen, sulfur kind of aromas will be produced, and it also the growth of the yeast cells will also get reduced. Next one, it also slows the fermentation. If the sulfur aromas are more, sulfur compounds are more, that is DMSO, that is dimethyl sulfur oxide. So, this dimethyl sulfur oxide is travels from the beginning of the thing, like in malting stage itself, the DMSO starts traveling. So, this once it should be removed. So, if it is not removed, it slows the fermentation process, and it also poor venting, it gives the poor venting of fermented gas, and sulfur aromas will also correspond to the unnatural flavors will be produced by the yeast. So, the summarizing the overall talk, so the yeast which is present in beer corresponds to the flavor. So, it should be so this yeast gives the life to the beer, and in addition it also helps the properties like flocculation, wort fermentation, ethanol production, and the flavor of the beer is released out by the yeast cells. So, the characteristic flavor and aroma of the beer depends on the strain of the yeast which is used for the fermentation. Thank you.